Alright guys, here we have lesson 7 group problems, questions 1 and 2. So 1 is asking about the genome of 4 specific viruses, and it asks you to dive in a little bit to endonuclease versus exonuclease, one of which attaches to the end of the genome, which is the exonuclease, and the endonuclease attaches to the middle of the genome. So that's how we can differentiate between circular and linear. Linear, it'll attach to the end, and circular, it'll attach to the middle. So with that, we look at virus 1, which is single-stranded because here we have the A and the U, they're not the same, so they're actually single-stranded. And it's circular because it is resistant to exonuclease, but sensitive to endonuclease, and it's RNA because it has uracil instead of thymine. So virus 2, using the same topics, we can find out it's single-stranded as well because the probabilities don't match up on each nucleotide. And it's RNA because, again, it has uracil, and it's linear because it is sensitive to both. So genome of virus 3 is single-stranded with DNA because it has thymine this time, and it is linear. And virus 4 is single-stranded, circular, and DNA. So question 2 is the fill-in-the-blank section. So A, topoisomerase 2 is composed of 4 protein subunits and uses ATP to compact bacterial chromosomes, while a nucleosome contains 146 to 147 base pairs of DNA and 8 histone proteins. Remember those 4 main types of histones, they're 2 each. B, the DNA double helix is 2 nanometers wide. Adjacent base pairs are separated by 0.34 nanometers in the DNA double helix. C, topoisomerase 1 introduces positive supercoiling into DNA and cuts 1 DNA strand. The origin of replication in E. coli is called, I don't know how you say that, but it's ORIC. And E, the Hershey-Chase experiment, bacteriophage T2 proteins were labeled with a radioactive sulfur-35 isotope, while bacteriophage T2 DNA was labeled with a radioactive phosphorus-32 isotope. And the sugar in uridine is RNA nucleoside, while the sugar in deoxythymidine is DNA nucleoside. All right, thanks guys.
This is James Leo. I'm doing problems 3 and 4 for the lesson 7 group problems. 3A asks us, how long is the DNA portion of this translocation chromosome in meters? To find that, we first combine the 2 chromosomes together, which will give us 157 million base pairs. Divide that by 10 and times that by 3.4, since that's the length in between each base pair, and that should give us 4.618 million nanometers, which is equal to 0.004618 meters. Question B asks us how many nucleosomes are found within this translocation chromosome. Assume that the average length of the linker DNA is 50 base pairs. So we know that there is 147 base pairs of DNA in each nucleosome core particle. So in order to find out, we divide that by 157 million, and we multiply that by 50 for the linker, and that should give us 53,401,360.54 is the answer. For question C, we are asked how many H2A molecules would you expect to find within this translocation chromosome. We just used what we got previously, multiplied that by 8 because that's how many histones there are, and that should give us around 8 million H2A. Finally, it asks us for D, how many radical loop domains would you expect to find within this translocation chromosome? So for loop domain, we know that there's usually, I believe, let's see. Yeah, 25 to 200,000 base pairs of DNA per each loop. So that should give us around 785 to 6,280 loop domains. So for question D, for question number 4, it asks us if the chromosome was stretched out linearly, how long would it be in micrometers? So just to calculate that, we use 0.34 nanometers first to get around 1.4 million nanometers, and that's equivalent to 1,499.4 micrometers. For part B, it asks us what's the diameter of M tuberculosis chromosome in micrometers. So to get that, we divide that by 3.14, and that should give us 477.5 micrometers. And for C, it's 0.34. It asks us if the loop was stretched out, which it contains about 50,000 base pairs, how long would it be in micrometers? So to calculate that, we use 0.34 times that by 50,000 base pairs, and that should give us 17,000 micrometers. And lastly, it asks us how many histones are found attached to M tuberculosis chromosome, and we use, so to get that, we use 4.4 million divided by 147 base pairs, and that should give us 30,000, and we multiply that by 8, and that should give us 240,000 histones.
So I'm in charge of problem number 5, Dylan, and it gives you different molecules A through F, and then at the bottom it asks you a couple questions. So the first one is which of the molecules in parts A through F are nucleosides, and so the part B is also with nucleotides, and the difference there is we're looking for phosphate groups, so we can see that the ones that don't have a phosphate group would be F, E, and A, and so that's what I have down there. And then the ones that do have phosphate groups, which are B, C, and D, those would be our nucleotides. So then we're talking about ribose and deoxyribose in the next part, and that is based on the hydroxyl groups that you have. So deoxyribose is going to have 2 hydroxyl groups. So ribose is going to have 2 hydroxyl groups. So we can see that F has 2, E has 2, D only has 1, and C has 2, and then the rest of them only have 1. So for my ribose, I put C, E, and F. And then deoxyribose was going to have a single hydroxyl group, so that's when you have A, B, and D is the last one, and that's what I have for my deoxyribose. And then the last part talks about pyrimidines, and this is all based on the ring structure. So purines are the ones that have the double ring structure. So we can see that this has a single, so F, E, D, C, those are going to be our pyrimidines because B has the double ring structure, as does A. A. So that's problem 5.